Thank you. Thanks for the organizer for the opportunity to come here. It's very interesting, first time for me in Russia, and it's a very interesting experience. Now, first of all, my talk is going to be very different from the talk so far because it's not going to be about bioinformatics education. The other thing is, because there are a lot of actually bioinformaticians in the audience, I feel kind of awkward because I was told to target my talk to undergraduate biologists. So please excuse the watered down version in terms of technical details. Okay, so what I'm going to talk about is estimating statistical significance of sequence motifs. And in particular, what I'm interested in is in the de novo motif finding. And I'll strictly talking about DNA, although pretty much most of what I'm going to say, if not all, is apply for amino acid sequences as well. So as you all, I'm sure, know, motif finding is important for the identification of cis-regulatory elements, in particular transcription factor binding sites. And it's a fact that many tools were and still are developed for de novo motif finding. And by de novo motif finding, I mean a tool that would search for short sequence motifs given only a set of input sequences without any additional information. Notable examples that I've listed a few. I just want to point out that this is pronounced meme, not meme or mimi or something like that, which is the European version of it. Consensus, GIP samplers, and there's quite a bit more there. Just for the off chance that some of you might not know what de novo motif finding look like. So this is a cartoon of how it might look like. You're given a set of input sequences. So here in this example, each row is an input sequence. And under the model we'll be considering, you assume that each of those sequences have an instance of the motif. And you're asked to find these common transcription factor binding site or any other motif. So what do? You press a button, and voila, the motif is there. So I don't know, I mean, the colors, if you can see them, but the sites are now highlighted in pink, and these are the common site. This is the motif. Now, real life is, well, the problem is a little bit more complicated than that. The problem is that these sites are not exactly the same. If that was the case, then a repeat, like a suffix tree we've just seen, would easily solve that problem, but it's not. These sites are different from one another. They have some common theme through it, which is where the name motif comes from, right? So it's a common theme, but it's not exact repetition. So here the consensus sequence was highlighted by capitalizing the consensus letters in each position. And it's this variability from the consensus that makes this problem a little more challenging, both algorithmically and in terms of my perspective of significance. Now, I'm not gonna, this is as far as algorithms for motif finding go. We'll just press a button as in this talk. And really my perspective is that the current tools for de novo motif finding are really good enough. And as an editor, I get a lot of new de novo motif finding tools, and I think it's wasted energy from my perspective. And it's not just because I have a tool. So what I'm going to talk about is related to understanding the output of those tools. So a typical motif finder output would look something like that. The finder would produce the alignment of the sites, that the collection of sites that would be all taken together is considered the motif. So in this particular example, I have an alignment of depth 5, so 5 sites, and the width is 13. That's the number of letters per site. Now, this alignment gives you all the story you need, all you need to know about the motif, but sometimes all you need to know is too much, so we look for a way to summarize the information. And one common way to summarize it is by looking at the so-called profile, or PWM, position weight matrix, or position frequency matrix. And what this gives you is simply some kind of a statistical summary of the alignment. So for example, the first column here, you see 1C and 4 T's. So you see the first column here, there's 1C and 4 T's. In a position frequency matrix or a weight matrix, this might be translated to 0.2 here and 0.8 here, which is the relative frequency of these counts. So that's one way to summarize the alignment, and already we lost some information by this summary, but it's much more manageable to work with. But even this is too much information, because if we are given that, then, you know, and then we are asked to find what's the significance, it's going to be difficult. So, and coming to the notion of significance, and really that's, as I said, that's what really interests me in this problem, is the motif finders that come in many flavors, and they almost invariably give you something. It almost never happens that motif finder says, "Oh, sorry, couldn't find anything." They always find something. And despite of the proliferation of the finders, which I told you still comes about one a day, the significance analysis, the statistical analysis of the results have been somewhat neglected. So many finders just give you the result and, you know, there you go. Not anything about the significance of it. Others, including some notable ones, actually don't give you the right, the right significance analysis, even as what they state it is. So they state something and then they calculate the wrong thing. Yet, from my perspective, I'm now in the statistics department, a reliable significance analysis is an invaluable asset when it comes to decide whether you want to carry on with the reported motif to the next stage of the analysis, be it a computational or a biological analysis. So, how do we go about assessing the significance of a motif, right? So how can we assess the significance of a sequence motif reported by a finder? That's the question I'll be talking to for the rest of this discussion. So you might have heard about the entropy score or the information content. That goes back to Gary Stormer, who had a lot of positive contribution on this, fundamental contribution to this area. And so what it is, this is the expression. Here is, I'm detailing all what you need to know. If you've seen it before, you've seen it. If you haven't, don't panic, not yet. What it is, it's simply a summation over all the, so remember W is the number of columns in the motif. That's the width or the length of the motif. And you're summing up the contribution for each column. What is that contribution? It's basically a measurement, this measurement right here, the inner sum, is called the Kuhlbeck-Leibler distance. Never mind, it's just a name. But what it really is, it's a measure of the distance between the background frequency model and the observed frequency table of that column. So each column has its own frequency table, if you remember from that position weight matrix. And then you can ask, well, how diverged is this observed frequency table of that column is from the background. So basically what the entropy score is giving us, or the information content, is a measurement of how far is the motif from the background. The further it is, the happier we are. Okay? So the entropy is commonly used as the target function for the finder optimization process. So a typical finder would, for example, try to optimize the entropy distance between the candidate motif and the background. So while it can do that, the entropy score cannot compare alignment of different widths or depths because of normalization issues. And more importantly, it does not indicate how surprised are we by the reported alignment. And it really is that measure, that surprise level, that really amounts to the statistical significance, right? So when we ask for statistical significance, essentially we're asking for how surprised are we at what we observe. Let's just look at a simple example. We're given a coin which we believe is fair, so that is our null hypothesis. And we are told that this coin was flipped 100 times, and 75 times it landed on heads. Okay? So the question is this surprising to the point where we become suspicious of the coin's presumed fairness, of the null assumption? And how shall we quantify this? So statisticians often do that by reporting the p-value of the result. So what's the p-value? It's simply the probability, assuming the null assumption is correct, that this is a fair coin, of observing a result as extreme or more than the one we saw. So in this coin example, because under the null assumption, this is a fair coin, the probability of heads is 1/2. We have 100 tosses, so we have 100 independent trials with the probability of success in each one is 1/2. That's a binomial random variable, and we are asking, well, what's the probability that this binomial random variable had 75 successes or more? So, so that's the question, that's the p-value. Now, translating this to the language of motif finding, we would suggest this: the p-value of a reported motif would be the probability of finding such a motif or better by chance. And for this to make sense, we need to agree on what is the null model, how are random sequences generated. So the first thing we would probably agree on is that null sampled input set should have the same dimension as the original set, and by that I mean same number of sequences and same length. How technically would we sample them? Ideally, we would sample from, say, genomic sequences, or from a genomic train Markov chain of order 3 or 4. That's ideally. However, typically those are intractable if you want to do some math. So we might have to settle for an IID or zero order Markov model for sampling genomics, for sampling these null sets. Finally, we're asking, well, what's the probability of finding such a motif or better? So we need to somehow linearly rank the motifs, and one way we might do that is using the entropy score, which we've seen before. Okay, so now we have some idea of the what we need, and now it turns out that even under this simplified IID null model, we don't know how to compute the p-value of a motif with entropy score S, and by that I mean, what is the probability that a null drawn input set will contain a motif with score greater or equal to S? That's a problem we don't know how to solve. So when I say we don't know how to solve, we don't know how to solve it practically. So therefore, Hertz and Stormore suggested adopting the BLAS notion of an e-value to solve this problem. So in order to understand how to do that, so first you identify a motif as a motif with the alignment of its sites. So that we already have gone through that. Then the e-value of a motif with score S is the expected. That's E for expected. The expected number of such alignments, so by such alignments I mean alignments of the given depth and width as the one I've seen reported by the finder. The expected number of such alignments that score above S, assuming the input sequences are generated according to the null model. Okay? So that's pretty much the same definition of an e-value that BLAS uses, and everybody uses BLAS e-values. They're reliable, they're tested. And the reason that BLAS reports e-values rather than p-values is not because they don't know how to compute. They don't know how to compute the p-values. They do. The reason is here, this quote I've taken from Altshull, Stephen Altshull, is that e-values of 5 and 10 are more understandable than the corresponding p-values, which are not particularly telling, illuminating. And moreover, when the e-value is small, the p-value and e-values are nearly identical. So there doesn't seem to be any loss in talking about e-value rather than p-value in the BLAST context. So that seemed like a very reasonable idea, especially since I don't know how to compute p-value, or we don't know how to compute p-value, and e-values, I'll show you that we can compute. The e-value is the expected number of motifs or alignments that score above S. Therefore, it's simply the product of the number of alignment in the input set, which is a huge number, times the probability that a single random alignment of the same width and depth as the reported alignment will score above S. And that's a very small number. It's written minuscule there. And now, conceptually, this probability, this latter probability, is easy to compute. It's, okay, so a little technical detail. The probability mass function of a single random alignment of W columns is given by the W-fold convolution of the probability mass function of the entropy of each column. So for the mathematician in the audience, this would make sense. For the others, don't worry about it. It's not critical. All I'm saying is, mathematically, it's a simple problem. Mathematics aside and reality aside, if you actually want to compute this efficiently and accurately, which you need because it's a very small number, then that's actually challenging. And it's challenging to the point when that's where I got interested in this problem, is that we showed that the current implementation of this thing, so the e-value is reported by both mean and consensus, but both of them actually are not accurately computing this probability. In particular, mean can be orders of magnitude off. So it can give you an e-value of 1,000,000, yet the true answer is 1. So why is that? So I won't go into the details, but just very high-level explanation of what's going on. So, okay, so I need to compute probabilities. So how do I compute probabilities? There are 3 ways I can think of. One way is to use exact formula. So in our simple coin example, the binomial, I simply sum up. I want to know what's the probability that I'll see more than 75 successes. So I sum up over all values more than 75. I sum up the probability of the binomial random variable attaining that value. So that's using exact formulas. I can use Monte Carlo simulation. I will repeatedly flip the coin 100 times, and I'll observe the proportion of tests where I observed more than 75 hits. So that's the second way I can think of. The third way I can think of is using asymptotic results, such as extreme value theory or central limit theory. And in this case, I would say use the central limit theorem, which tells me that I can approximate a binomial distribution with a normal one. And this is just using the standard normal approximation to the binomial distribution. So these are the 3 ways I can think of. The first 2 ways, exact formula and Monte Carlo simulation, were ruled out as too slow for the purpose of consensus and mean. So they use the third one. But there is a problem with the third one in the context of motif finding. And the problem is this, that we can occasionally obtain motifs which score close to the maximal possible value. And that's exactly where the asymptotic results actually tend to do poorly, when you're getting really close to the maximum possible value. And just to show you an example, this is the normal approximation to the binomial probability distribution. So the x-axis is simply the number of possible successes from 0 to 100. The y-axis of each point, the y level, is simply the log 10 of the ratio between the correct probability and the normal approximation. So you see here in the middle, the expected value is of course 50. So around 50, you get that the ratio is 1, log is 0, which says that the approximation is accurate. But notice what happens towards the ends, the maximal value. It's 7 orders of magnitude off. So it's extremely poor approximation, and that's pretty much a common theme. Asymptotics don't work at the extreme values. Okay. So that's the first problem I got me interested in this, and I worked.
Question.
Yeah.
In practice, what would constitute an extreme value? How to find this threshold where approximation stops?
Okay, so that's a good question, and that's really what most statistics books gloss over. They just tell you, use the normal approximation, and then they give you these, what I consider ridiculous guidelines, like, okay, if it's above 20 or above 40, you're fine. But they don't say, well, yeah, if the number of trials is about 40, but what's the number of successes? So it's really the ratio of the number of successes to the number of trials. And yeah, it's typically not, there's no rule of thumb that I can give you that right off the top of my head. But you really need to look at the problem like what I did here. Okay. So that was the first problem I got interested in. So can I solve this problem? I mean, can I get an accurate calculation of the e-value? So we spent on this quite a bit of time, and we came up with actually a new technique that allowed us to do the first approach here, to do use exact formula, but use it but in a way that's sufficiently fast that is acceptable for incorporation into a motif finder. So we solved that problem, and very happy, we have a way to compute fast and accurate e-values, and then what did we learn? E-values are not the right thing to look at. Why? Let me give you specifically what's the example we looked at. So example we looked at constructed 400 Twilight Zone data sets. So these are 400 data sets that were constructed, drawn, so each one has say 20 or 30, I don't remember exactly, sequences, and they were fairly long sequences, and they were drawn according to an IID model to make sure that the IID assumption wasn't violated, because the e-value uses an IID assumption. Then what we did is we implanted these sequences, these random sequences with Motif, which was just barely enough above the noise level. So just barely. If a good Motif finder would find it, a lot of others would find some other junk or would struggle finding it. And our GIP sampler successfully recovered the implanted Motif in 288 of these 400 data sets. That's great, but here comes the surprise. The median e-value was 10 to the 12. So in other words, again, 288 successful runs of the Motif finder. It finds the right motif. Compute the e-value, and I computed correctly, I know that. The e-value comes up as 10 to the 12. The expected number of random alignments, the score above what you found is 10 to the 12. And yet you found the right motif. Okay, so two things. The lesson from here is twofold. One is e-value is not the right thing to look at, because nobody in their right mind would accept something with an e-value of 10 to the 12. The other thing is, what is going on here that my motif finder, using some apparent paranormal resources, is able to find a motif yet among this huge haystack, right? Okay, so our motif is not psychic, motif finder. But that's another question which I can't go into right now. It's actually an interesting question. How come it's finding it? But the bottom line is, do not use e-values. Okay. Well, okay, so the only good news out of it is that the method we worked hard to develop actually can be applied to other problems. So it wasn't a complete waste of time computing these e-values correctly. Okay. So why is the e-value failing in our context where it's such a success in BLAST? Nobody doubts BLAST's e-values. Recall what Alciu said. when the e-value is small, the p-value and e-values are nearly identical. And that's correct for the pairwise alignment problem, since the number of high-scoring maximal local alignments is well approximated by a Poisson distribution. So in case of a Poisson distribution, the probability that you'll observe at least one, right, when that's the p-value, is given by 1 minus e to the minus lambda, where lambda is the parameter of the Poisson. And for small lambda, that's approximately the same as lambda. And lambda, of course, is the expected number of the distribution. So yes, when lambda is small, for the pairwise alignment problem, the p-value and e-value are about the same. Not so for the motif finding context, because the problem there is that the number of high-scoring alignments does not seem to follow a Poisson distribution. It is much clumpier than that, the data. That is, if you have one motif, one alignment that's score above your threshold, then typically you'd have a lot of satellites. Because if, say, that alignment has 30 sites, if you replace one of those, you'll still have high-scoring alignments. So what happens is that once you have one above alignment, there's bunch of them just jumps in, so it's really very different from Poisson. And what it means is that the e-value might be 10, but the p-value is might be very, very small, unlike what you have in the pairwise alignment. So the idea of using an e-value seemed reasonable at the time, but in hindsight, no, it just doesn't work. Okay, so let's go back to the p-value, and okay, we don't know how to compute it, but still, is it useful, regardless of the fact that we can't compute it efficiently? So this is the next question we asked. So we looked back at that 400 Twilight Zone data sets that I told you about, and these 288 successful runs. And the question we asked is, if we use the p-value, which is simply the probability that the input set will contain an alignment that score above the reported score, S, how many of those will be significant? So it turns out that at most 10% of these successful runs will be declared significant at the canonical 0.05 significant level. And that means, remember that you would like as many of these 288 to be declared significant, because those are all, I know the implant, I know these are successful runs. I just want to know, can it be declared significant? And only 10% would be declared significant. So that's way too conservative using the p-value. I say that I don't know how to compute it, so how did I actually know that 10% will be declared significant? Well, I don't know how to compute it, but I can still estimate a lower bound on it, and I won't go into that. So that's an accurate assessment there. Okay, so why is the p-value also failing? Okay, we understood why the e-value is failing. Why is the p-value also failing in this context? And to understand that, let's go back to the coin example. And in the coin example, imagine that we don't actually see the number of heads. Rather, we have some absent-minded reporter who counts the number of heads. And that person, about 20% of the times, might miss the heads. So he might report 60 heads rather than 75. In that case, the p-value will drop by, or increase by 5 orders of magnitude, from roughly 3 times 10 to the minus 7 to 3 times 10 to the minus 2. Huge difference in terms of the significance. The problem is that unlike BLAST is near optimal, or if not optimal. Motif finders are not anywhere near optimal. So they're like these absent-minded observers. They give you some result, which is nowhere near the top. So, and that's the problem here. Okay? So the p-values they report, the p-value based on what the motif finder gives you is a lot higher than if that motif finder would have been an oracle that would have been able to pick the really best motif. So what, yeah?
You told us in the beginning that motif finders are already close to the limit of perfection. How possible?
I didn't say that. I said they're good enough for what we need. right? So, and there's a big difference. In these twilight zone searches, my motif finders were successful. They could have been better in terms of optimizing really the entropy score. They could have been much better than that. But who cares? All I care is that I'm close enough to the motif, right? That's really the essence of it. Yes, you can do a lot better optimizing there locally, but it doesn't really matter.
So are you saying that your motif finder actually find like consensus type correct profile?
It finds a profile which is close enough to the, which if you look at it and you look at the right one, yes, it's obvious that they found the right thing. But yeah, right, it's not the optimal one. So, so we need to take the performance of the finder into account when evaluating the statistical significance. And whereas, okay, so let me just say, how can we do that? So we call that finder-specific significance analysis. And I use this notation of a finder f together with the specific parameters that you have given that finder. That's this f. Is applied to a data D, and f of d is the score of the optimal motif that finder with these parameters found on the data D. If I knew this distribution of f of d for a null-drawn data set D, then I could report a finder-specific p-value, which is the following, which is what is the probability under the null model. Under the null model, the data is generated according to our null generation model. What's the probability that the finder applied to this random data set will exceed the observed score of S? And notice the big difference from before. Before I ask, what's the probability that the data set contains a motif of score S? Now I'm asking, what's the probability that the finder will find something greater than S? That difference is what makes one p-value wrong to look at from our perspective, and the other is the right to look at. Okay. Okay, that would work, except how do I find this finder-specific distribution? Right? The problem is there are practically unlimited number of combinations of finder/dataset parameters, right? Every time you change the number of sequences, their length, their composition maybe, that changes the graph, that changes the distribution. So that means we have to generate this finder-specific distribution on demand. But the problem is, I have no idea how to do it theoretically, right? So that's the only way I know how to do things fast on demand. If I have some theoretical formulas, I don't. So what do I do? So the only thing I can do is rely on some ad hoc computational intensive Monte Carlo simulation. So to create a sample of n points, n scores, what I need to do is draw n independent data sets according to the null model. Okay, that I can do fast. But then I need to apply the finder to each one of those, and that's going to take a while. So in practice, that means that I typically have to limit my sample null distribution to about 50. Okay, so that's like a ballpark estimate, though. That's what we actually use in practice. And that means that, yes, I drew before this picture with this really nice smooth graph of the distribution. You can't get this kind of smooth graph with 50 points. Right? So it's not going to be anywhere nice as that. Still, under the null assumption, the score itself, the observed score itself, came from the same distribution as these 50 sample points. So I can join it in, and I have 51 or n plus 1 points. And I can report the p-value relative to the empirical distribution based on these n plus 1 points. Right? And as an aside, I would say that's the same thing as noting the rank of the observed score in the combined list of n plus 1 scores, the n null scores and the observed score. So for example, if my observed score is higher than all the 50 other null scores, then my p-values will be 1 over 51. The good news is this is a non-parametric method, that it's robust. I'm not making any assumption about the score's null distribution. The bad news is rather coarse. The best p-value I can hope for is like 1/51, which is really not going to cut it for, especially if we're considering several data sets where we might have to deal with multiple hypothesis testing. So we would like to get a little bit more than a p-value of 0.02 essentially. Or if we're comparing competing motifs, looking for the best one, 0.02 doesn't allow you to work with much. There's no flexibility there. So, okay, but we're stuck with that. That's what we can do unless the null distribution is well approximated by a known parametric family. In this case, what I can do is I only need to estimate the, say, 2 or 3 parameters of the distribution rather than the entire function. I don't have to estimate a nice smooth graph, just have to estimate 2, 3 numbers and then derive the graph based on those 2, 3 numbers. Okay, that would work, but which parametric family? Others have assumed extreme value distribution and normal. So we looked at it from fresh. And this just shows you a couple of examples. What you see here are probability plots where basically I take a parametric family and I tweak the parameters, I estimate the parameters so that the data would sit as nice on a straight line as possible. These look almost like they've cooked up because you see how well the data fits the straight line. I swear it's not cooked up. These are 2 different finders. This is our finder. This is meme. 2 different models of null generation. This is IID model. This is sampling from genomic sequences. So actually sampling the data from sequences. That's the ideal kind of null model from where I come from. The only surprise was I didn't tell you which is the parametric family. Normal, EVD, neither. It turns out, and this is something that I just stumbled on, 3-parameter gamma. So what is the 3-parameter gamma? You take a gamma distribution, you add, which has 2 parameters, shape and scale, and you add a 3rd parameter, which is location. And those 3 parameters then allows you to have these wonderful feats. Okay, 3 parameters gamma. And this is just to show you that the data really doesn't follow a normal distribution. The green curve is the best fit you'd have normal. The purple is 3 gamma. You see the problem with the normal. It's nowhere near normal. Okay, so estimating the parameters. We estimate the parameters of the 3 gamma using maximum likelihood estimation. That yields what we call the consistent point estimator of the p-value. So what we do is we estimate the 3 parameters gamma, we draw the curve corresponding to these 3 parameters, and then we look what is the p-value relative to that curve. And it's nice that it's a consistent estimator, which means that as the sample size grows, the estimator will converge to the correct p-value. But for any finite n, there could be considerable variation in the estimated value. And here's what it looks like. So in this particular case, I took a gamma distribution. And the correct gamma distribution, the real null gamma distribution, is the blue curve right here. And I'm trying to estimate the p-value of 60, and the real p-value is 0.046. That's the tail of the blue distribution. And the other 3 curves you see here were based on drawing samples from this null distribution of size 50 and estimating the parameters of the distribution, and then plotting those curves. And what you see is while the red and the black one are reasonably close, the green one is already quite a bit off the correct. Okay, so it's not 6 orders of magnitude like we had with the eval, but it's still more off than what we would have liked. Can we do something about this? Yes, we can. When estimating a parameter, we often face this problem of variability because of the sampling process. And how we control for that is using confidence intervals. So in our case, the parameter is the p-value of S. How am I doing with the time? Do I need to? Who's the 5 minutes? Okay. So yes, we can construct confidence intervals. Never mind the technical details. And this gives me this confidence interval. Because it's a confidence interval, of course, it has to be a conservative estimate of the real p-value. And that's what we report. Oops, conservative. I threw out e-value and the previous p-value, which was based on the probability that the dataset contains an alignment rather than the finder can find an alignment of greater or equal to. I threw those out based on the fact that they were too conservative. Now you're giving me another one which is conservative? Well, like in the Republican Party, there is conservative and there is conservative. So let's compare quickly the e-value and the p-value with the conservative estimate I just introduced. So recall again the 400 Twilight Zone searches data sets I mentioned before. 288 of these were successful runs. The median e-value, I remind you, was 10 to the 12, certainly way too conservative. All of these would be thrown out. Using that p-value, that theoretical p-value, at most 10% of these motifs would be below the canonical 0.05% threshold. Using the newest p-value I just demonstrated, using this finder-specific estimation and confidence interval, now instead of less than 10%, I have almost 60% of these motifs are below the significance level of 0.05, so they will be declared as significant. So it's still not 100%, but it is conservative, but it's a lot better than 10%. Okay. At the same time, about 7.5% of the 112 runs where the motif finder was not successful, only 7.5% of those were below the 0.05 significance threshold. So it's pretty good. The best answer would have been 5%. I'm a little bit above 7.5%. There is an explanation for that. I don't have time to go into it. Let me just say that aside from this, the fact that it actually works, there's huge advantage to using Monte Carlo simulation is with the e-value, I was stuck with the IID model. The IID model is really not good for DNA sequences. With the Monte Carlo simulation, I can use whatever null model I want. I can adjust the null model to factor in any property of the input set which I considered inherent to the input set but might inadvertently have an effect on motif finding. For example, we show that local GC content might have a substantial effect on the ability to find junk motif and their score. So that's something you might want to consider factoring in, and we were able to do that. Okay, let me skip that. Let me just tell you what it is. It's basically I showed you that I can estimate the significance of motif as a post-processing. But if you have a good estimator of significance, you can now start using it to choose the right motif between several candidates. So that's what this is about. I'll skip it. Finally, so thanks to the people who worked with me on this. So my two former PhD students, Neil Jones, who was the same Jones from the Jones and Pevsner book. He was very prolific. So he also worked with me on this. Pavel, who was my mentor going into bioinformatics. And this is kind of a memorial thing because since moving to Sydney, grants have not been forthcoming. So I missed those. Okay, thank you. Yes.
So there are two related questions. One is the issue of false positive. Suppose you don't plant a motif. Would you still occasionally find something significant?
Yeah.
And the other thing is that on the level, on significance level of 0.5%, of 5%, you would have in your 400 runs about 20 significant motifs by chance, by definition of significance at that level.
Yeah, right. Okay, so if they are related, then yes. So let's go back here. Right. So the thing is, okay, so here's why I got this 7.5% and why. So remember here, there were 288 successful runs. So when I'm estimating false positive, those 288 are out of the window. So there only remains 112. And on those, I had 7.5%, which is higher than the 5% I was aiming for. And the reason it's higher than the 5% I was aiming for is because those are random motifs that were found by the motif finder, even though there is a real implant motif there. So in other words, once you condition on the fact that these are high-scoring random motif finders, bets are off. And that's why it's 7.5% rather than 5%. If this was really 400 null data sets, then it would have been 5% or less. In fact, it would have been less because I'm doing a conservative estimation. Yes?
Two questions. First, you started by saying that you are talking on a biology undergraduate level. Did you keep it all the way? What kind of
no.
value do you ever see?
Okay, so... Okay, so I did mention a few mathematical details, but I did say that those are for only people who are interested, and I think you could have followed the talk without needing those details. That was my intention. Maybe I aimed too high still.
No, I enjoyed it, but I think
thank you.
biologists
Might not.
might have got lost. But
Well, is there a biologist in the crowd? Yes, okay, so, were you lost?
It's good.
But just one biologist, and this is what I was aiming for? Okay.
And another comment actually is to defend the motif finder industry, of which there are about 200 by now.
Yeah, this is accepted. There's like a zillion which were rejected.
So I think part of the reason that there are so many is that the problem is very elegant and easy to
Yes.
solve. And part of it is that none is very good. If
Pardon?
there was one great motif finder, there will not be produced so many more. And another comment, the problem is changing, because by now we have, for example, protein binding microarray data, which pose completely different problems.
Right.
You could play with the same ideas, but they are completely
Yeah, of course. When the problem is changing, I have no problem. But because of Pavel and his challenge problem, I still get tons of paper for review with people trying to solve the challenge problem. Forget it, you know, from my perspective, this is solved. You don't need that. I mean, yes, I agree. With new problems, sure, there is room for.
The challenge problem is also too artificial in the sense that the motif is actually a cave.
I agree. Yeah, I agree. That's why I don't like these papers. But they come in all the time. They still come in. You have an enormous effect on on these people. I mean, and yes, I agree. It's because it's elegant. So it's elegant. People like it. Oh, let's try this idea. They solve it. They send it for publication.
I have also a question. Given a large-scale online bioinformatics course and your lecture as a part of this course, for example, what programming assignments or questions do you like to see during your lecture? I mean, to make the learning more active.
If I would ask people for that particular aspect, so it depends who you target. A computer scientist, you'd probably give. Okay, yeah. So first of all, I would give them some question about algorithm. I completely disregarded algorithmic issues, but it's not because I don't think they're important. It's just because I think it's well developed enough. But going over doing some understanding the algorithms behind motif findings, I think is important. And I would probably give people Monte Carlo simulations, like the ones that we did when we discovered all these things. I think it would be fascinating for people to actually do this kind of discovery by themselves, to play with the e-value and see that it's just not working, you know, yeah.
So, given the, you know, if you had this problem in real life, in real, you know, you have a real biological problem, you want to find, what would you recommend as a combination of tools in this setup? So, no RNAs, no
yeah, de novo motif finding, you say, yeah, okay. So, I really like our own motif finder. It's Gim-san. The reason I like it, it's a bit slowish, but it really scrapes the barrel. And I can show you just very briefly. We did, okay, I won't show you. But we did some comparison with a data set, a real biological data set, and it was way better than all other de novo motif finder in that test that we compared against. The downside, it's a little slow, but the advantage is it gives you a reliable significance analysis based on what I just told you. At the moment, I'm not aware of any other significance analysis that is as reliable as ours. We are in the process of incorporating this into MIME. So MIME would be hopefully within the year would have the same kind of significance analysis. But yeah, so from my perspective, pardon?
MIME is slowish already.
MIME is slow. Well, okay, depending on what your data set. If your data set is chip-seq, yes, it's slow. But if your data set is like 30, 40, up to 100 sequences, then it's fast as far as I can. I mean, pardon?
These are the exception now. The recent data sets are much larger.
Yeah. Okay. So this for chip-seq data, these are different types of questions. Yeah. And their MIME is definitely slow, and in fact, Tim is working on a much faster version. Yeah. Thank you.